Welcome to this webinar. I'm Enrico Guarini, faculty member at the University of Milano Bicocca, and today I will be hosting this event with my colleague Francesca Magli. We thank you, all of you, for taking time at your very busy schedule to participate. This educational webinar is entitled Data and Resource Management in Research Infrastructure. And today you will be hearing from our guest speaker, Dr. Paul Flicek, Associate Director of EMBL EBI Services, with responsibility for data resources and related activities. He's Senior Scientist and Head of Genes, Genomes, and Variation Services at EMBL EBI. This webinar is part of our Executive Master in Management of Research Infrastructure and Executive Education Program of the University of Milano Bicocca and Right Train and EU-funded Horizon 2020 projects. Some detail about our webinar. Today's agenda includes a lecture by Paul, followed by a Q&A session in which our guest speaker will answer to your questions. Of course, a recording will be available after the webinar. And to request to speak, click the raise hand icon that appears next to your name. I see. Sorry,
Jan and Kim and another have to close the microphone.
Yeah, exactly.
Okay.
You hear a noise. Let's continue. Sorry for that.
Kim, also, please. Okay.
Please unmute all your microphones, please. Yes? Mute all. Okay. Sorry. Okay. Today's agenda includes a lecture followed by a Q&A session in which our guest speaker will answer to your questions. Recording will be available after the webinar. So to request to speak, click the raise hand icon that appears next to your name or use the chat features. Okay. Now it's my great pleasure to introduce our guest speaker, Paul Flicek. Paul, first of all, thank you for sharing your knowledge with us. Paul has a long experience of setting up and managing data resources and information within research infrastructure. Paul has been an EMBL senior scientist since 2011 and team leader for vertebrate genomics at EMBL EBI since 2007. His team develops the ensemble genome annotation resources and analysis infrastructure. And also Paul has worked on aspects of genome annotation, comparative genomics, and the large-scale biological data generation projects for more than 2 decades. So, okay, it's time to start. Paul, please, the floor is yours.
Okay. Thank you very much, and thank you everyone to your patience as we had some of the technical difficulties. I hope that today's lecture is interesting and useful for all of you. So just to give you an outline of what I plan to talk about today, we'll have a very brief introduction, and then a small discussion about data management plans, fair data principles, and then an example of data coordination in a distributed project and how this is done in detail. I've decided to present a detailed example because I think this gives, it covers a lot of the issues that are relevant here and really gives a sense of how we do things for data and resource management. I'll then have a few words about the GDPR and other legal considerations at the end. So to start with, data and resource management is really all about sharing. That's it, both within projects, so not necessarily sharing with the wider world when it's in the context of running a project, but also for those cases where it is sharing with the wider world, with the wider community and the wider scientific community. And of course, as science has become increasingly data intensive, there's been a recognition of the role of effective data management, and this continues to be a major theme going forward. My experience, as Enrico mentioned, is from genomics and big data projects in biology. There's various challenges in these projects, but they fall into a number of categories: data volume, data complexity, and logistical complexity. And so we're going to cover all of those in greater or lesser extent as we go along. And so, Jin, also just a brief comment on data and resource management plans. These are now often required by funding agencies at the time of application, and it's a very common deliverable in EC-funded projects that a data management plan will be one of the early deliverables. But regardless of whether it's required, it should be considered as an early piece of basically all relevant projects. That is, all projects that are developing data and resources. And although format specifics vary based on the funding agency or something else, a data management plan which addresses each of the FAIR data principles and then describes relevant details for the expected data and resources is generally the best approach to produce something that's consistent and understandable across all of the people that will be affected by it. So, of course, why share data? The first question, the answer to this that many people will give you is because you have to, because the journal or the funder requires it. But I think there's actually quite a number of better reasons conceptually, in that sharing data accelerates research, it increases citation rates and the standing in the community. It maximizes value to basically who the people who have funded it, be that public or charities, increases the impact of the research and fosters collaboration. It also stops wasteful duplication of data and promotes innovation via downstream and integrative analyses. And many examples of all of these cases could be given. So I'm going to spend a little time then talking about the FAIR data principles. This is something that was formalized just a few years ago in 2016 in a publication that described them. And it's a way to formalize and standardize many of the existing practices that had been happening in the community at the time. It also builds on the experience of a number of foundational resources from a number of areas of science, including resources we have here at the EBI, like Uniprot or Ensemble, or resources that exist in other domains of science as well. And it has the advantage of providing a way for funders and others to benchmark the result of our data management activities, which, again, I think is a valuable role that these principles have. So I'm just going to briefly note the principles. I assume that a number of you will have seen these already. So the first of these is findable. And this is really about assigning persistent identifiers, describing the data with rich metadata so that its context and its meaning is clear, and to make sure that there is connections between the metadata and the data that they describe, and to make sure that these metadata and the data are indexed in a searchable resource so that there are identifiers with metadata that can be found. For accessibility, the keys here are to make sure that data can be found using standard methods. So standard communications protocol, very often this will be searching over the internet, but there may be other reasons to associate with this, and that the protocol allows for authentication and authorization where necessary. For example, when dealing with ethically sensitive data or commercially sensitive data. In addition, that the metadata remain accessible even if the data no longer are, so that it's possible to find out what the data was actually about, these concepts. Data being interoperable means that they are represented in common languages and in vocabularies that follow the FAIR principles. I'll talk about this more in the detailed example, and that they are referenced to other appropriate data and metadata where required, so that there's an interconnected way that the data and the metadata are represented. And finally, reusable. Reusable data is basically make sure that the data have attributes that make it clear how it can be used. So clear and accessible data usage license. This is actually a critical aspect of this. Much data that exists is either not licensed or not noted as to what the license is, or there are conflicting or confusing licenses. The data are associated with provenance, so where they come from, how they were developed. This is especially clear when data resources are built from multiple underlying data resources, and that they meet the relevant community standards. In this way, that as data is generated in different parts of the community, it can be fit together in a coherent manner. So those are the basics of the FAIR data principles. These are very common, talked about almost all the time. I think it's important to consider that they are, for most resources, still aspirational, in that it is very rare to find a perfect resource, in that the data meets all of the various pieces. And it is a challenge, although many of the long-standing resources meet a large number of these various principles. So I'm going to talk through now a relatively detailed example of how we manage data in practice and how we apply the FAIR principles in practice. And this is about making functional annotation data useful and accessible. And the project that I want to focus on is something called the functional annotation of animal genomes. So this is a global project. It goes by the acronym, or the pronounceable acronym, FANG. And the goal here is to identify functional elements in animal genomes and to create a rich genome to phenome resource. There's a number of key aspects to doing this, and you will recognize them as very relevant to the FAIR principles. Standardizing metadata descriptions, core assays and experimental protocols, coordinating and facilitating data sharing, establishing infrastructure for data analysis, and then finally, the overall goal is to provide this high-quality functional annotation of animal genomes. So this is a project that we've been involved with at the EBI. It's a project that, although it has worldwide partners, there have been 3 new European contributions that started last year. They're focused on different aspects of animal genomes. The first, AquaFang, on aquaculture species, the second on chicken and pigs, and the third on cattle. These represent, although are not all of the species that are covered by the worldwide project, these are a fair representation of the type of species that this project seeks to collect and integrate data from. Here at EBI, we run the FANG Data Coordination Center. This is a way to make this data organized in a way that it can be easily reused, and to make sure that these various pieces of the FAIR data principles are applied, so that the data is richly described, consistently reported, standardized a protocols and analysis method, and then facilitates general openness, reusability, and so forth. So the key questions here are what's needed to make research data sets useful to others. In the context of the FANG project, there's a few things. This is just an example of the types of things that we want to collect. One is information about the samples, so the species, the breed, where the data is from, how old it is, which tissue, what protocols are used. And another is about experimental assays. One of the major experimental assays in the FANG project is something called ChIP-seq, which maps protein DNA interactions in genomes. And so here there's key questions of the experimental assay that are required to ensure that data generated in multiple locations and from multiple samples can be effectively analyzed together. And I've just listed some of those that are available, that are required here. So in FANG, we've developed a metadata, or there is a FANG metadata and data sharing committee that is set out to help coordinate all of this. And it's put together so far 118 different metadata questions, which are broken down into sample attributes and experimental attributes. Not everyone who provides a data set needs to provide answers to all of these questions, but there's there's a flow of questions based on the various samples or the various experiments that take place. All of this is described in a concept that we call a rule set, which I'll describe in a little more detail. But again, to harken back to the FAIR principles, to describe these data sets, we want to make sure that we capture enough information so that the experimental data set is fully understood. It could be repeated based on the information that we have, and then easily reused in the context of other data sets that have been generated by other people. All of this requires the use of consistent terminology and, of course, be well structured. And here's just an example of the type of information that's collected for ChIP-seq analysis of histone modifications, which builds on the experimental attributes that I described a couple of slides ago. So, of course, both biology, but here biology as a field, not necessarily biology as a concept. Biology evolves, specifically the technologies associated with experimental assays. And because of this, we update the rules that we use to describe it. The FANG rule sets are now on version 3.8. It's taken us 25 releases to get there. And these describe new technology types, as well as improvements to our understanding of what's important. So one of the key aspects of data management is this flexibility and the ability to update things, but maintain a version control system so that one can look back and understand what the rule set that was used 2 years ago or 4 years ago when data was generated and provided into the primary resource at that point. We version control our rule sets using GitHub, which was originally designed for managing software development, but is now used in many situations where you want to have a version control of something that you are doing. So past versions remain accessible. And then we have a pieces of software that describe them as human readable tables, which is an example just shown on the bottom of this slide. So, of course, why do we really need good metadata? The key point of this is to make data usable, to reduce ambiguity, to facilitate reproducibility, and again, to improve the integration across the labs and projects. We also want to make data discoverable, not just by other resources, but by the major services that are agnostic to the specific projects, but want to pick up the metadata, pick up the data, and use it in an automated way. And having good and consistent metadata makes this possible. And of course, it improves analysis. It makes it easier to potentially find batch effects or confounding factors when we've captured information about how the batches or the experimental protocols were put together. But of course, these rule sets are only useful if they're followed. It's not at all uncommon that a very nice set of rules are put together, but the rules and the SOPs are not necessarily followed. And so therefore, the way to make this work is to validate and curate these data sets against the rule sets. For large numbers of data, it's impossible to do this curation by hand. And so automated tools are required to facilitate validation against the rule set. And this is just an example of the types of things that would get caught. Here, there's metadata associated with 4 data sets. And if you look across this, you will immediately see that there's a whole host of inconsistencies in the various metadata associated with the sample. Now here the data might be well, you can see in the final column, the data is actually DNA sequencing from a specific assay type. But what you can see is that there's multiple types of error that need to be fixed or checked, from different names to refer to the same species, different ways that sex is represented, dates of birth, sites associated with sample collection dates, and so on. For all of these things, it's relatively easy for a human to determine, for example, how to compare a collection date that's written as 27-October-14 versus 02/01/2012. The human mind can do that, but a computer sees these as very different ways. Similarly, a computer will see an assay type type of RNA-seq with a capital S and RNA-seq with also lowercase s as two different assay types. And so we need to make all of these things consistent. So when it comes to things like describing aspects of the biology, we use a tool, a very specific tool to describe information. In biology, these are ontologies. Ontologies are unambiguous ways to describe biological entities, and it provides a short description that then is reflective of the larger description. So here's an example that we use specifically in the FANG project. This is about endocrine system disease. There's a whole host of synonyms that are associated with this that may appear in various different databases. And when we collected this information from databases, that's where we got this list of synonyms. All of this is then described by a single unified term, endocrine system disease, and this identifier at the bottom, EFO, which EFO stands for experimental factor ontology, and then a specific number. And once we use this, we then have a common terminology that we can use across all of our data sets to make sense of the final data. And ontologies have another characteristic, which is that of hierarchy. And so each item in the ontology knows its place in the global worldview. So for example, in the hierarchy that's shown on the right of this slide, you can see that if we are describing tissues, we can start with animal, whole body, head, brain, different parts of the brain, and each one of those boxes that has a plus will expand into further details into the hierarchy. And so people can then select information from various parts of the hierarchy. If you're interested in all data from the brain, you can select at the brain node. But if you're interested in only data from, for example, the brain stem, you would select deeper into the hierarchy and find information that way. So it's also true that we don't, of our 118 different metadata, different questions, not all of this is created equal. Some fields are tagged as mandatory, which are always required, and validation will fail if we don't have them. Others are recommended in that it should be provided, and the validation will fail unless it's specifically described in a specific way, as in not applicable or not provided. And then there's a final set that are optional. These would be useful, but are not required to pass validation. And so you can see down at the bottom, there's different types of information that is absolutely required. In this example, sex is required. Birth date is something that is recommended. So using these concepts, the ontologies, the metadata model, and the rule set, we've put together a coherent data model for the FANG project, where we go from organism to sample type to, organism to specimens to cultures and cell lines. And then each piece of data is associated with all of this metadata and can be tracked together in that regard. There's an important question as to how to best collect data from the community. If you remember from the world map at the start of the description of the FANG project, there are data generators all across the world on every continent. And so one of the easiest ways to do this is to collect data via spreadsheets, which are accessible to all, and easily passed between lab members. It can also be automated in various ways. But a spreadsheet, if you've worked with one of these, they're easy to make changes on such that they have inconsistencies. And so that's why we have developed the FANG metadata validation service, which requires people to upload the completed spreadsheet and to select which rule or sample or which sample or experiment rule set to test against. And then there will be a report from this that describes the validation result. The data that passes, the data that has warnings associated with it, or the metadata that passes, the metadata that has warnings associated with it, and the metadata that fails, and the reason why it fails. This provides a way for those people who are generating data in the community and its associated metadata to know exactly where they stand, to be able to put together things that when incorporated into the main data set, will meet the FAIR principles and be able to be analyzed in an integrated way. Now, it's not always feasible to have this validation result viewed on the screen. Some people want to interact with this in programmatic ways. So they upload, for example, a CSV file, a comma-separated file, and then they get back an automatic result from our validation service. And so we can see this in the JSON format, which is a data format that is computer readable, easily computer readable, which is in the black box there. So there's an automated way to go from the metadata to the validation of the metadata tool. It's also possible to look through the output tab, which is a way to spot things like naming errors and other details, where you can look across to see, for example, how many different types of values exist in birth date down at the bottom, where there's multiple things that are shown. So our validation tools, then here's an example where the validation result has passed the metadata. It does a number of things, most of which I've described or alluded to earlier in this presentation, where it checks the ontologies, it checks various relationships that would be expected in the data, and it checks the minimum standards and that those standards are valid. All of this makes archiving the final data submission easier. So once the data has passed the metadata validation, it's ready to be archived, and then the archiving would take place potentially at a publicly available resource. For the example of FANG, we use the European Nucleotide Archive, and our metadata submission tool for FANG converts the metadata format into the correct format, thing that can be uploaded directly with the associated data to the final archive. And so this is the conversion for archive submission. Once we have the final validated metadata, it is then output in the appropriate format that might be submitted to two databases, in this example at EMBL-EBI, the Biosamples database or the European Nucleotide Archive database. And so we end up in a situation where metadata has been collected by users, validated by our system, and then submission files output. And data is submitted to Biosamples in the one case with one form of output, and then to the sequence data to the European Nucleotide Archive. And it generally needs to be done in this order, so that the sample data is registered in Biosamples, and then the experimental assay data is submitted to the European Nucleotide Archive, which points back to the sample data held in the Biosample database. All of this is connected via the FANG metadata rule sets and the FANG validation service. Now, of course, the European Nucleotide Archive and indeed Biosamples are very flexible databases. So they are not in a position to validate the information that would be coming from the ENA, or that would be coming to them from the FANG project, because they are supporting many projects, which is why this initial step is required for FANG, such that other FANG members will be able to access the data from these centralized resources directly. This is just an example of the experiment data associated with the ChIP-seq experiment and other experimental assays completed with the standard metadata. And here's just an example of the type of metadata that comes out and what it looks like. One of the things that we do with the data that ends up then in the ENA is that we create a data portal for all of the data associated with the project from all of the worldwide data producers. This is driven completely on the data that exists in the archive, which was shaped by the metadata portal. So data, again, comes from the community, goes through the metadata validator, submitted to the main archives, comes out of the archives into a central portal that then can be accessed by anyone and filtered with all of the rich metadata that we know is available on these samples, because we were involved in the validation of the original metadata submission. So to sum up this metadata section, talking about what makes a good metadata system, controlled vocabulary with consistent ontologies, minimal data requirements, encourage engagement and completion of the data, support and ease of entering data and instructions, batch upload and programmatic access, and validation to prevent information and errors. At this point, I have additional slides to talk about GDPR. I'm conscious of the time associated with this. I can go on. There's only 3 or 4 slides associated with the GDPR. I could talk about those, or I could take some questions at this point, or indeed I could jump directly to the summary. I'm kind of happy and at your disposal at this point. I don't know if I can see the chat window. Yeah.
Well, it's on the left side.
Okay.
Your thumbnails. Move the mouse on your left hand side of the screen, and then you see the pointer and thumbnails.
Yes, I guess it doesn't appear to me that there's any information in the chat window, but if there is,
I see one from Nicolas Casadei.
Okay.
Nicolas, do you want to make a question? Francesca, could you please unmute Nicolas?
Nicholas is already on mute.
Yeah,
Yeah.
he's muted. He's muted. But
Okay. Sorry. Nicholas?
Yeah, I was saying would just be glad to continue. Was quite good flow.
Okay.
Okay.
So in that case, I will spend just a couple of moments talking about legal considerations and the GDPR. Now, of course, the entire example I just gave was about farm animal data where the GDPR doesn't apply. Similar things, of course, have been and could be developed for data where the GDPR does apply. I noticed that there were a number of questions in the expectations about GDPR. Unfortunately, I'm not going to be able to answer all questions about GDPR. If I could do that, I would probably have a consultancy service that only answered questions about GDPR. But what is true, and we have worked extensively at EBI with GDPR, it's an evolution and extension of data protection in Europe with an emphasis on transparency, process, and governance. That said, most of the GDPR requirements were present in the directive from 1996, just without the levels of transparency, process, and governance that the GDPR envisioned. One of the complications with the GDPR that I think a number of you are, if you have engaged with this, will have come across is that although it is a regulation, the member states, in the case of this regulation, have significant flexibility to legislate. And this flexibility will almost certainly create diversity in how data protection is managed across Europe. And this diversity will be a challenge. There are many questions and lots of uncertainties and processes being developed. There's a number of resources that exist on the web. One of the ones that I've found most effective is from the Bird & Bird law firm, which is an international law firm who have been quite active in putting up information. I have the website down at the bottom. Information, and we've worked with them directly on aspects of developing data protection in some of the large-scale international projects that we're working on. So there's 2 major documents that have recently been released about the GDPR and scientific research. So one of these is shown here with the URL at the bottom. This is a roughly 100-page study about the GDPR. I think the quote at the top is an interesting and potentially an understatement. We found that the GDPR is not universally perceived as an enabler of research. I think that's very true. Many people see it as a barrier to research. Again, because of some of these ambiguities and uncertainties as to how it applies. There's a number of pieces of information in this document about how to address that. And so the entirety of this study, which again, as I mentioned, goes on approximately 100 pages, about options to evolve ambiguities and address procedural concerns. And indeed, how scientific research can find common ground that it needs to move things forward. More recently, in fact, just released last month, was a preliminary reading on opinion on data science, on data protection and scientific research from the European Data Protection Supervisor. This document has a number of very interesting aspects about it, and if you haven't taken the time to read it, I would recommend that you do so. One of the concerns, it's clear that comes from this document from the European Data Protection Supervisor, is the concern of essentially sham research taking place in commercial organizations to skirt some of the rules of the GDPR under the research provisions. And so there's an aspect of that discussion that takes place. But there's also a recognition, which is in the second quote that I've pulled out here, about how the term consent as a legal basis for processing in the GDPR has created confusion amongst research organizations. So consent as a legal basis for processing is conceptually and operationally different from informed consent that human participants in research have given. So one of the important points of this and of the GDPR generally is that research-informed consent is something that is separate from consent as a legal basis for processing. So it can still be the case that in some situations you can have informed consent, but you are using public interest or some other legal basis for your processing. This is an area that is evolving and there's still a number of questions associated with it. But this confusion around consent, I think it's very important that this has been recognized explicitly in this preliminary opinion. So the key points, I think, to take away from interacting with the GDPR is the GDPR provides many ways to share and process data. And I think focusing on the options for sharing is a much more effective way forward than a focus on perceived restrictions. So many of the discussions that we've had basically have started with the concept that the GDPR puts restrictions on things. I think it's a much easier discussion to have if you start by saying the GDPR gives us all of these options, and then we need to figure out which one of them are the most appropriate. It will still be the case in research that bilateral and multilateral data access and data management agreements will remain a key tool to facilitate compliance, privacy, and IP management. All of this we have used in the past. And there may be differences in the details in the context of GDPR, but they will remain a key tool. And it's, of course, this landscape is going to continue to change for many years. And the general advice that I would have and that we have in the projects we've been involved with is to seek advice when appropriate and to make justifiable decisions. And I think the GDPR helps facilitate this even with its current situation. And so to summarize, data and resource management plans should be based on FAIR principles. Data management and coordination in practice should connect project-specific requirements, like the ones I described for the FANG project, into existing reference databases, such as those of the EBI whenever possible, and to use these back and forth. Standards for data, metadata, and ontologies should be developed openly, so in a way that other people can see them and comment on them, and then validated with appropriate tools. And of course, the GDPR and related national legislation will likely require many changes to the details of data management, But I think most of the concepts of how we, especially in science and in research, use and share data, the concepts are well established. It's the details that we need to work out in the content. So that wraps up my talk. I just want to acknowledge Peter Harrison for aspects of how we do FANG. I'm happy to answer questions through the rest of the time. I see there is one question about long-term sustainability of data resources and services. So I think this is certainly a challenge. One of the ways that it can be addressed is to basically, as one of my summary points made, interact with the existing reference databases. Most of the reference databases we have at MLEBI, but there are others as well, have in general, although obviously nothing is guaranteed for all time, but have a longer sustainability horizon, and interacting with those provides one aspect of how data can be made sustainable by essentially making it the responsibility of people who are experts and funded to do that. More local sustainability, I think, is a challenge. Many tools or many data sets get generated in projects that have a definite end. Many of the EC-funded projects have a definite end associated with them. Thinking about that sustainability from the start is often very important. My experience with EC funding is that there is often relatively little direct sustainability for a specific project. And so how key data resources can be incorporated into a consolidation resource is a potential effort for that. Another big challenge for sustainability, especially resources, is for software that's been developed. I don't think we have very good solutions for that at the moment. Although it's clear that very important and widely used pieces of software do get maintained, I think we do miss and lose software that could be important, but just takes time to build up its user base. So this is an area that is a challenge. It's an area that I think we're getting better at as people come to realize more sustainability is required. The development of the FAIR principles have been an advantage here because it's a way to benchmark things and describe them with a widely recognized term.
Thank you, Paul, for your interesting presentation, and also for providing examples, concrete example of data management, and also references for the GDPR. I was wondering if you can talk a little bit about some managerial issues related to managed data services, because of course you mentioned about controlling, engaging people, validation in coding. And this, of course, is mostly related to the project manager level. Looking at different teams, and so the perspective of the managers of the research infrastructure, especially for distributed nodes, if you can make some suggestion on that, how to, what about the process of setting up specific guidelines and standards for data management and quality of information on that?
So I think setting up the standards is best done kind of from an outcome basis where people agree on what the ideal outcome is and then work backwards to determine what's required to get to that outcome. The more engagement that can be had on determining what the outcome is, I think the more successful they are likely to be. So the agreement on the outcome should be as wide as possible. But then I think charging of a representative or setting up a representative committee who is responsible for developing the standards to get to that outcome is a very effective approach. So wide agreement on the final outcome, but then a relatively small group that sets up the standards that facilitates that happening. I do think it's very important that the group that's working on this does so in an open way. My experience in some of these projects is that people tend to ignore all of this work when it's going well, or they think the direction of it is going well. And the concern tends to arise when it's uncertain what's happening, or when the standards are being set without a clear reason for the standards. If I can just give an example from a different project that I was involved in the 1,000 Genomes Project, which roughly 12 years ago started out to sequence many human genomes. At that time, groups had come in and set up standards associated with how one should store sequence data, but they were not about solving any problems. They were about developing a standard. When the 1,000 Genomes Project set out to actually do the analysis on the data, they found that those existing standards didn't work, and they had to create new ones that solved the problems that they had. And so this is where I get this idea that the standards should be problem-led, rather than the standards being led by kind of a description of the world or some other subjective term. The more that a standard can be problem-led, the more likely it will be to be adopted.
Thank you, Paul, for answering that. I see one question from Sashil. Please, Sashil, you want to make a question? Paul? Sushil? No?
Yes. So I was just reading the question from Sushil. This is about the European Data Strategy White Paper. And I have not seen that white paper yet. So I think the key, and it's about how do people contribute to a legislative framework for data access and empowering people with relevant skills to create common European data spaces. So I think this is an area that, of course, people associated with research infrastructures can give a lot to, because research infrastructures are almost always about these interconnections across countries and sharing data. Creating common European data spaces will require aspects of harmonization in the legal frameworks. The GDPR does provide an aspect of this, but the national legislation has the potential to make this harmonization challenging. So I think we need to provide examples, and this is an aspect that I think the EBI will also engage with, to provide examples of where harmonization is critical to pushing research agendas forward.
Thank you, Paul. I think we have time for one more question. Monique, please, you want to speak?
Yes, can you hear me?
Yes, please.
Okay, no, actually, I'm just posting on the chat some questions from Sushil because he has problems in typing or posting questions in the chat. So
Okay.
These are not from me, but I'm just copy pasting his questions for you, Paul, to answer. Thank you.
Yeah. So one of the questions is, at the EBI, do we have data management or governance frameworks, toolkits that can be reused? And asking, are the processes for making data fair themselves? The answer is yes, in that, well, it is our absolute effort and commitment to do so. All of the software we develop on the service side of the EBI is made available with a permissive Apache 2.0 license, so it can be incorporated into other tools for any purpose. We are, of course, very happy. We do provide code and examples and work with people in direct collaboration to do these various processes. These things are, of course, challenging to maintain as viable and flexible toolkits. And so we currently don't have a dedicated team, for example, that's developing toolkits to do this. But the processes that we do have are all available. So and then there's another question about a framework to measure data flows and estimate the utility of data. How can you measure the economic value of a data set and hence make investments appropriate to its utility? This is another question that we've been addressing at the EBI. There is we have a report that's available on our website under, I think it's under our impact, but it should be linked from the homepage. That was commissioned a few years ago about basically the value of EBI data sets using the type of economic analysis that governments often use when they are trying to determine if some other piece of infrastructure is appropriate value, for example, a roadway or bridges and things like that. And those analyses are often done by determining how it's used, who uses it, how that usage drives other usages further on. These results suggest that those data sets that are highly used and are sole source, so generally large data sets, have tremendous value. It's true that the data sets that are most valued tend to be reference data sets, and so the ones that are reused most often. How something becomes a reference data set is not easy to guess, although sometimes you can make the guess in advance. The human genome sequence was always going to become a reference data set, and because of that, it has high economic value. It's also encouraged a lot of investment. But I think we should also be comfortable with the fact that research has a broad distribution. There will be some data sets of very high value, some data sets that have less value but are still important for us to maintain because of their use for reproducibility. And some data sets, the value only becomes apparent further into the future. But so there are techniques that we have used. We have mostly borrowed the techniques that exist in other for measuring the economic value of other pieces of infrastructure. And then there's a question about FAIR as a qualitative measure. Do you have a way to measure the level of FAIR and recommendations as to how to improve the fairness of data? Currently, there are efforts underway to do this, and there have been some publications that have come out that have rated different resources on how many aspects of the FAIR principles that they meet. It is true that there are aspects of FAIR data that are subjective, and I think that's I think it will be impossible to be fully objective about many of these things because FAIR is meant to cross various fields. In more specific fields, I think you can get to a much more quantitative and objective measure. I think there's an analogy to be drawn to the example of how I described the metadata around the FANG project versus the generalized metadata that's associated with submission to, say, the biosamples or the ENA databases. The FAIR principles themselves have to be general to be widely applicable, but within certain fields, I think it would be appropriate to come up with stronger definitions. And the FAIR principles support these, where you have the FAIR principles about domain relevant community standards. This is something that could be within a given domain very directly stored as to whether or not the data would be available in that. Similarly, whether the identifiers are appropriately connected in searchable resources that make sense, and whether the language and the vocabularies are the ones that are used by the field. Those could be actually quite directly stored. And so then the final question is my whole personal genome personal data? This is a great question, actually. It's hard to know some of these things. There's an argument to be made that the GDPR, depending on how you read it, outlaws things like memoirs. Because when you're writing about a memoir, you're including personal information about people who are not you, your family, for example, and you may be doing so without their consent. So that's an interesting aspect to that. I don't think that's what the GDPR is meant to do. There's also an aspect of the GDPR that allows for data to be completely released under a manifestly made public concept. Unfortunately, manifestly made public doesn't have a legal definition that I'm aware of. So although you can make data available under that form, it's not clear how you would do so. So it's an interesting question. There are aspects of the GDPR that deal with genetic data as a specific data type, but I think really what all of this comes down to is consent. The human genome sequence, which is fully open, does come from individual people, but I don't think there's an argument that that's personal data of the same level of something that you never meant to share. So an interesting time that we live in. I think it's my experience with the GDPR has been intellectually very stimulating, although some parts of it have been frustrating to nail down the details in practice.
Okay, I think time is expired. This is very interesting answering a presentation. We got a lot of questions today. This means that the topic is very mainstream and interesting. And so thank you for your questions to Paul. I think if you have additional questions on this discussion, you can send to us. And I think Paul will be glad to answer again. And we will use the master platform to answer to specific questions. I think now it's time to go. Okay, thanks so much, everyone, for joining us. And of course, a special thank to you, Paul, for sharing your experience with us. Thank you so much, everybody, and have a great day.